Thanks. My name is Stefan and I'm studying genetics. And today we'll hopefully present to you the idea of using bacteria as sustainable factories to produce different chemicals. Probably you're not familiar with the idea of biotechnology, but the concept is fairly simple. You just need to give instructions of what you're interested in to bacteria and just give them food, and they happily, willingly produce more or less what you've told them to. This is the type of bacteria that I was working on is Cyanobacteria, which means that they have this process called photosynthesis. As you're familiar with, it's like in plants, they only require sunlight, water, and air in order to grow, replicate, and produce all of those useful chemicals. So here is a very simple concept of the idea how it's applied into the industry. You need to have different types of containers that grow the bacteria in. They need to be exposed in sunlight so that they can take the energy out of the sun, and then you just basically take those bacteria and filter and kind of take only the good stuff that you're interested in. The species that I was working on is, as I said, type of cyanobacteria, which is just basically a group of cyanobacteria that perform photosynthesis. Its name is Synechococcus 7002. It has been chosen because it has the fastest growth rate within the cyanobacterium, which is useful when you want them to produce something. As I said, it uses photosynthesis, which is the biggest advantage because when you compare this to the current standard system that have been used in biotechnologies, it's E. coli, when you've heard of it, and E. coli needs fresh water and sugars. So you actually need to grow plants, take sugars, feed them, and then they can produce what you're interested in. Whereas cyanobacteria, they only need filtered seawater because they're a marine organism, sunlight, and air, carbon dioxide from the air. So it really don't need that much input, and they can already naturally produce a wide range of chemicals, such as a variety of pigments. Some have applications in cosmetics as like sun protection cream, biodegradable plastics. You can already imagine the application of plastics everywhere, and probably most importantly, medicine with antibacterial functions. Some have reported to have like suppressor actions on tumors, so that's quite important. However, the biggest setback so far is the fact that the process of taking the DNA into the cell, giving them the instructions that they need to make those chemicals is quite inefficient for this type of bacteria so far. So this is where my project comes in, when I was trying to identify factors that affect this so-called natural transformation, because it happens naturally within the cells, and then combine any beneficial factors and see if I can improve the efficiency of transformation. This is a basically very basic layout of the process. It's fairly simple. You just need to grow the bacteria in liquid, as I said, because they're a marine organism. They just grow in seawater. You take some of it out, you add the DNA on top of them, just like you can mix sugar with tea, and incubate them for 24 hours, which will allow them to actually read the instructions and produce what you're interested in. And finally, count the colonies that have been successfully transformed, which is basically the measurement of success in the transformation reaction. The more colonies you have, the better the transformation was. So here at the beginning of my project, I was interested to see if the culture of the stage will have some effect on this. And an easy way to think of culture stage is different stages of development. At the beginning, they're like kids, then they grow into like adulthood, and at the end, the later into their stage they're like elder people. And with these different stages, they have like different properties, which supposedly might have an effect on transformation. So this was the first thing that I looked at, and then the amount of DNA, because the instructions we give them and always in the form of DNA. So if we give them more or less, would that change the transformation? And finally, the period of incubation. I was interested to see if we can like keep it for longer. They'll better understand what we're trying to do with them. So here is my first set of data. I know it looks a bit weird, but I'll explain. On the y-axis, we have the number of transformed colonies, which in this case vary from 0 to 12. I should say that in E. coli, the transformation, when it's really good efficiency, it yields about 1,000 transformed colonies, and here we're talking about from 0 to 12. So it's really low efficient, as you can see. Here at the bottom, I've used optical density, which is just a measurement of culture stage. The lower the optical density, the earlier into their development they are, and the bigger the optical density, the later into the development they are. You can see here, this is the established protocol, which was the protocol that was established by the time I started doing my job, and it uses fairly early cells from an early stage of 1 optical density. I have repeated the experiments twice, repeat 1 and 2, and they're marked with X and O's, as you can see. And the take-home message from this graph is that almost with none of the new optical densities or culture stages, there was any improvement into the transformation efficiency. You can only see like 1 out of the 2 experiments with 3 transformed colonies, which is still quite low, and even the current protocol only worked once and yielded 1 colony. So the transformation efficiency is very low and does not improve with culture stage for this particular experiments. Then I went on to see about the amount of DNA, and this is the same familiar graph as before, with the only difference that it's on the x-axis this time is only amount of DNA used in micrograms. And again, the important thing here is to know that while increasing the amount of DNA, so the more instructions you give them, they actually a larger proportion of them start to get and understand and be successfully transformed in a sense. Although there is a difference between the 2 lines, the important thing is that they're all going upwards with increasing DNA, and they increase the transformation of the colonies. So the take-home message is that increased DNA improves transformation efficiency. Finally, going to the incubation time, fairly simple as the previous graphs. Here I've changed the incubation, which is the current protocol was 1 day of incubation, and I've made it up to 2 days and 3 days. And although it's not quite certain about 2 days, the data is not that obvious, but for 3 days, I managed to get the most transformed colonies, which is a positive thing. So now I have identified that longer incubation and more DNA actually helps us transform those cells. What I did next was combine those factors and generated 2 combinations, as you can see here. This stands for combination 1 and combination 2. And I've compared the number of average transformed colonies from at least 4 replicants. This time I managed to repeat the experiments 4 times. And for the established protocol, as you can see, which used 1 microgram of DNA and incubated for 1 day, I managed to get about 1 cell out of 4 experiments, which is 0.1 on average. So it's still quite low. However, with combination 1, the average this time was around 120, and for the combination 2 is even more, about 240, which still says that there is still place for improvement. If you can add more DNA up until this stage, it still makes more transformants. And hopefully, very high transformation efficiency is now achieved, which is something that you can easily now work with. And some of the implications of this are that because recently, up until now, the process was really low efficient, and researchers, if they wanted to start and work with this organism, has these amazing capabilities, weren't kind of avoiding it, because if it's only going to work 1 every 4 times, you really don't want to start doing any experiments with it. But now, because it's way more efficient and it works every time, and it produces like hundreds more cells than initially, hopefully this will encourage more research and students and labs to actually use this and exploit and develop the mechanisms that it has to produce different chemicals, which will probably lead to have a higher production from the same amount of cells, which could probably be implicated in different emerging sustainable industries, because, as I said, it doesn't require anything but sunlight, water, and air, which is abundant everywhere, even in the less developed countries. All you need to invest is just some kind of a container that will keep them together, and they're readily starting to produce interesting chemicals that you can sell and use. And eventually how this affects us will be that we'll have available on the market cheaper and eco-friendly in sense products such as all of the derivatives of plastics, because once you can produce plastics, you can have all of these applications. Everything that comes like in color because of all the pigments, in cosmetics, hopefully will be found some useful applications within the antibacterial pharmaceuticals, because you know that's quite a really problem currently because of resistance to antibiotics. And they can also produce alternative sources of energy such as ethanol and hydrogen. And most importantly, they produce these amazing chemicals by taking up the carbon dioxide from the atmosphere. They take molecules from the atmosphere and then combine them into those chemicals that I've previously mentioned, which is very interesting because and important because, you know, carbon dioxide emissions are the main problem for climate change and pollution and carbon footprint in general. So by producing all of those useful chemicals, they'll also manage to kind of lower the footprint that we have. And here you can find some of the references that I've used, and I would like to thank everybody in the lab, which was Professor Ampman, that I took my research into. And of course, thank you for listening, and if you have any questions, I'll answer them now.
Just out of interest, what kind of number of colonies do you need in a sample to kind of expand that out and then make that into more industrial applications outside the lab?
Well, you only need around 20 successful colonies, because once you have a successful colony, you only use them, you won't use the unsuccessful ones. So as long as you pick a number high enough that you can make sure that these are really what you're interested in, and then once you've selected them, they can grow, and everything else from then will be transformed, because they'll be kind of the kids of the successfully transformed colony, so everything else will be successfully transformed as well, which is, you basically don't need more than you don't need to go up to thousands of hundreds like E. coli does, but up to 20, it's good. I mean, it'll still do the job.
And then they're good for the foreseeable future. They don't like change or die off.
Yeah, well, no, you can always freeze them in a freezer up in minus 80 degrees, and they can last like this up to 80 years. So even after 80 years, you can always come back and just take some of them, revive them. That's the process called reviving, and they can still be alive and viable and go for generations to go, like years and months.
Thank you for that. I was wondering, would you expect diminishing returns if you, for example, put in 30 micrograms of DNA and left it for 4 days? Or like, was there a reason that you stopped at 3 days? Or do you expect that to just get better and better in terms of success?
Okay, I presume there will be a point that even if you give it more DNA, it won't be able to take it up, because it has like things on the outside of the cell that take up the DNA, and they have like a saturation. If you have 10 import channels and you had 10 DNAs, you'd take them all. But if you had 25, you'll still be able to take only 10. So as of this point, you could probably add more DNA and it will still affect it, or you can probably incubate it for longer, that will still have an effect on it. But I stopped there because of the project was only 30 days long, just the type of the honors project. And really, this data is really good enough, so you don't need to develop it anymore. Once you have 100 cells, you don't need to work more. It's the idea in biotech is that if it works, it's good. You don't need to make it better. Yeah.
Thank you.
Okay.
I have one more question.
Yeah.
What made you choose cyanobacteria?
Well, because of their properties, that I said, they're quite sustainable because they don't require anything compared to E. coli, and they're quite useful, easy to work with. They're probably the easiest organism to do natural transformation with. Other bacteria, you need to kind of give them cold, heat shock them again at 45 degrees for seconds and so on. Whereas these, you just add the DNA on top of them and that's all. They do all the rest of the job. So it's quite easy and sustainable, and it has already a wide range of applications.
Great, thank you.
I have a question. I'm going to exercise speaker's prerogative here. And you just mentioned that those guys, the cyanobacteria you used, were better for transformation than E. coli. I always found it really exciting to watch the expressions on my friends' faces when I said that I worked on E. coli in the lab, because the only time you hear about it, if you're not a scientist, is when there's an outbreak and it's killed people. But no, we have safe E. coli. So my question is, the DNA that you were putting in, what did it have the instructions for, or was it just a test to see if you could get the transformation step to work?
The one that I was working with was just a test, and he explored a fluorescent protein, GFP. So it means that if you give them the DNA, they'll start fluorescing green, emitting light, and you actually see that if they're green and emit light during nighttime when you just give them UV light, it means that the protein is here, which is native for, I think, a jellyfish. So it was just a type of a test, but then this could be always replaced with something more useful that we use in like different products and compounds. But only for the experimental part, it was just a marker, just to see that it works.
Okay, cool, thanks.